Good afternoon, everybody. Okay, so I will present the latest results in a project that has been going on since 2020. I've been working with Spike for quite some time now. So for those that are not familiar with the structure of Spike, it is a trimeric viral glycoprotein. It has more than one conformational state. It needs to open its receptor binding domains in order to bind to the receptor ACE2, and that is also a very important epitope for antibody recognition. And it is a highly glycosylated protein. So to cover a few of the background on this project, it started in 2020, mid-2020, once we evaluated the possible dynamical effects of the mutation D614G that appeared in Europe mid-2020. And we saw, according to our normal analysis methods, energy 10 more precisely, we saw that that particular mutation would make the closed state of Spike more flexible and the open state of Spike more rigid. And our hypothesis was that therefore it would make this Spike protein more prone to open and less prone to close, favoring the active state in which it could bind to the receptor ACE2. Based on this hypothesis, we modeled over 17,000 single mutations on those two states to see which other possible mutations would have the same effect. And we found quite some good candidates that had that effect even increased. We were already talking about doing some experimental validations of those candidates. But later that year, alpha appeared, and alpha was carrying one of our candidates, which was N501Y. And experiments have shown that mutation actually increased the open state occupancy of the spike protein. Beta and gamma were also carrying a few mutations that we predicted, mutations on K417, that we had many substitutions in that position that were among our candidates. But once delta appeared, none of the mutations that describe delta were among our candidates. So we started to realize that maybe other evolutionary pressure factors were playing a role in the evolution of spike that we would not be able to predict only looking at the conformational dynamics. Another thing that happened, I will talk precisely about when Omicron appeared. In the first 2 weeks, many structures were solved for Omicron, and different papers were describing those structures, their own structures, and pointing out possible different effects of the many mutations that describe Omicron on the receptor binding domain and the receptor binding motif. But they were pointing out not necessarily the same effects for those mutations. And it's not hard for us to understand why. Once we superpose those different structures, we see an RMSE of up to 8 angstroms for some residues in the receptor binding domain. So they were creating those evaluations based on snapshots of a protein that is not rigid. So based on those two things, why am I calling this a comprehensive structural analysis? Based on 2 aspects. So one of them is regarding functional aspects, because I am considering antibody recognition, receptor binding affinity, and glycan coating beyond the conformational dynamics that we have been evaluating since 2020. And also regarding the number of structures, we are working with over 1,500 structures of spike. Those are complexes, non-complexes, trimeric, peptides, domains, glycosylated, non-glycosylated, everything that was available in October 2nd last year. And we are able to do that because we are using methods that are suitable for high throughput applications. So I will first talk about our methods for evaluating interactions between spike and antibodies. So we use surfaces for that, which is a method that gives us a vector of interaction because it evaluates the pair residual interaction between 2 or more chains of a protein. And here, for example, you guys can see that this receptor binding domain has many different residues, and those residues have their net interactions with the light and the heavy chain of this particular antibody. We see even some interactions that are not favorable, which is kind of similar to what was discussed in many of those great talks about frustration. Then we align those vectors because a few of those structures are of peptides or of single domains, or they might have insertions or deletions. So we need to go through this aligning step. And we repeat that for all the data set. For this particular case, the data set of complexes with antibodies was of 942 structures. And we ended up with more than 2,000 vectors because a few of those structures are of trimeric spike or have more than one antibody bound to them. So we needed to cluster those antibodies, those vectors of interaction, sorry. And there are many ways to work with vectors, but we decided to go with a method that would consider both things that we are evaluating. So both the geometry of this interaction and also the strength of interaction per residual. So we chose to go with a weighted average of positions where the weights are exactly this per residual interaction. And clustering those, we ended up with 14 clusters. I don't know if anybody here works with Spike, but it is very well used, the 4 epitope definition by Bloom. So we ended up with much more than 4 epitopes. They are distributed all across Spike. Here are a few examples. So epitope 1, for example, is right on the C-terminal domain of Spike. Epitope 9, that is this green here, that is quite large as well, even though it's not even comparable to 3, 4, and 5. It's right on the tip of the N-terminal domain, and it's one of 5 epitopes in the N-terminal domain. This method also allows us to sort other vectors of interaction for new complexes or even for interactions with the receptor ACE2 among those clusters. So here it's a clustering of 146 structures, so 210 vectors of interaction between Spike and the receptor ACE2 among those clusters. And we see that the main cluster is the number 4, but also a little bit of number 5. And here we see exactly that the number 4 is precisely on the receptor binding motif, but 5 is a little bit different regarding the geometry of the interaction with the receptor binding domain, but quite close to the receptor binding motif, and also sharing a few residuals. We also did the evaluation regarding occupancy specificity, because those epitopes can be hidden or exposed depending on the occupancy. So for that, we did a similar approach, but considering the interactions between different spike chains for different conformations to see how those interactions would change depending on the conformation. And we also did the clustering for that. And here in green, you guys can see that parts of cluster 3, 4, and 5 are only exposed with 1 up or more receptor binding domains. But there is also a very interesting result, which is this purple here on cluster 3 that show epitopes that would only be available for 2 or more receptor binding domains open, which is another differentiation that the Bloom classification doesn't do. And here you guys can see exactly this epitope 3. And here, this image even show a sterical clash because we have for these reference positions of alignment only 1 receptor binding domain open, and it is having a sterical clash with this neighboring receptor binding domain. So regarding this occupancy specificity, exactly what we did for the modeled single mutations back in 2020, we also did now for experimentally solved structures, pairs of structures for different variants of Spike. And we see here something that we kind of hypothesized back in 2020 or 2021 once Delta appeared, which is an initial increase in open state occupancy, probably associated with increased transmissibility, increased ACE2 binding. But particularly since BA1, there's a decrease in this open state occupancy, probably associated with immunescape. And considering the previous results that we had for the single mutations that we modeled, we see that this is probably associated with one particular mutation that appeared for the Omicron subvariants, which is in the position 375. We also did evaluation of the glycan coating, but I think I'll have to go through that a little bit faster. And we had some very surprising results regarding the glycans, because we started looking at them thinking that they would be masking some epitopes. So they would contribute to immunescape by masking those epitopes and avoiding antibody recognition. But what we actually saw was some co-interactions between antibodies and Spike mediated by those glycans. Particularly on epitope 3, we see that the presence of those glycans can actually increase binding of the antibodies in up to 12.4%. I'm sorry. So this leads us to a trade-off hypothesis. So we are currently working on a manuscript that will describe this a little bit more, and I unfortunately will not have time to show some examples. But this trade-off hypothesis is based on the fact that those different functional aspects, they are connected, right? Because, for example, a same electronic interface is binding to some antibodies and to the receptor ACE2, so mutations there will change both. Some antibodies are recognizing positions that are only accessible in a few occupancies, or conformational states, so the conformational dynamic is impacting as well the antibody recognition. And regarding the glycan coating, those glycans can be also mediating conformational dynamics, and we have some interesting results on that I unfortunately will not be able to show as well. And they can be either masking or mediating interactions with antibodies and with the receptor ACE2. So take-home messages are that there is extensive structural information on spike, and that can be used if we have methods that are appropriate for high throughput applications. And this process can also be used for other proteins that we have a lot of structural information on. We are proposing a new data-driven approach to classify epitopes and also to sort new complexes among those classes of epitopes. We are pointing out that those different functional aspects of spike that can be driving the evolution are interdependent, and it's crucial for us exactly to understand this evolution and maybe to predict future variants, to understand those different functional aspects altogether. I would like to thank my team that I'm really lucky to work with, and also the collaborators. These 3 collaborators are co-authors in this work, all the institutions that made this research possible. Here are some references that I mentioned of previous publications of ours on this topic. These results are still not published, but they will probably have these names, so please check it out when they are. And these are the tools that I mentioned, surfaces for evaluating interfaces of interaction, and NRG10 is the normal mode analysis method for the dynamical evaluations. And thank you.
I have time for one question.
Yeah, it's a very nice study. So I was curious about the epitope regions that not only interact with antibodies, but also with the ACE2 protein. So are there some of these epitopes well understood, like from your structures, in the sense of like how do you handle their energetics when you're applying those weights in your epitopes?
So those weights are only for us to be able to cluster them together based on the position of the interaction. But we are seeing some interesting results regarding exactly those shared interfaces between antibodies and the receptor ACE2. Most of our results point out that a mutation would create the same effect on both. So many of the mutations that we are currently seeing, they decrease antibody recognition, but at the expense of decreasing ACE2 interaction as well. But yeah, this is an interesting thing to observe, because these point mutations also have different effects depending on the context that they appear. So for example, the N501 right in the beginning was increasing interaction with both antibodies and ACE2. But in the context of the latest subclasses of Omicron, this effect on increasing antibody recognition is decreasing because of this context of other mutations that are very close by. So it is a very complex interplay between those two different functional aspects of the same domain.